Hello everyone. So I'm going to talk about my research on the genetics of lipedema and kind of give you a flavor about how we're going to search for these genes underlying lipedema. And so the results that I'm going to show are still preliminary, and hopefully they'll be published soon. So I've got nothing to disclose. So first I wanted to talk about why we want to study the genetics of any trait or characteristic or disease. And I want to bring home the point that there's really two ways that genetic association studies or identifying genes can help us. One avenue is that identifying genes tells us about the biology of the disease. What are the tissues that are affected? Specifically, what are the tissues, what are the pathways, what are the molecules, the proteins. So understanding about that will maybe lead to better treatment and prevention. The other cool thing that genetics offers us is the ability to identify individuals, to say this individual is at high risk, and to be able to predict that risk and kind of personalize treatment plans according to their that person's genetic profile. So data from the UK Biobank recently emerged in the past couple years, and this was a great opportunity to look at the genetics of many different traits. So it's a very large study of 500,000 adults with extensive phenotypic or physical characteristics and genotype measurements. So the types of measurements that we might be interested in for lipedema include height and weight, waist and hip circumferences, and then body fat percentages in different parts of the body, as well as body fat percentages assessed from DEXA, which we've talked about. And we can look at the phenotypes in relation to the genotypes, so the genetic markers. And so the genetic markers that we're looking at are so-called SNPs, single nucleotide polymorphisms. This person has an A in their that specific code in their DNA. This person has a G instead. Does that difference associate with lipedema is basically the question, and we're going to be looking at that across the genome. So in the absence of information on actual diagnosis of lipedema, our thinking has been that we can use other information to identify women who likely have lipedema. And this information is looking at, on the x-axis there, you can see the mean leg fat percentage from this bioelectrical impedance measurement, where you stand on a scale and hold the sticks. And then also that in relation to waist circumference. So our thinking here is that the women most likely to have lipedema are the ones in the kind of southeast corner of that plot there. So the women who have a lot of fat in the leg in relation to a relatively small waist. And there's many ways to slice and dice this, and many potential ways to identify these women, but this is just one potential way. So I'm not going to get too much into the weeds here, but basically we identified, which you can see in green, the women with a relatively high leg fat percentage and a relatively low waist circumference. So basically our hypothesis here is that those women are likely to have lipedema or something resembling lipedema. And then we excluded some of these quote-unquote close calls in yellow. Then we did what's called the Genome-Wide Association Study, where we tested each of about 15 million variants in the genome in relation to the phenotype. So each dot here is a particular genetic variant, or SNP. One person has an A, other person has a G, all across the genome. On the x-axis is the 22 chromosomes and all the different spots along the genome. The genome is a very big place, and so there's many different variants we can look at. The higher up on the plot that SNP is, the stronger the association for with lipedema. And so you can see we kind of got a mix in this kind of first pass at it. Kind of got a mix. And here, another point that I didn't mention is that we're assuming a 5% prevalence And if I had time, I would love to poll the audience in terms of what they think is the prevalence of lipedema. But so I'm assuming a 5% prevalence, but I'm going to be trying it with different levels of prevalence, assumed prevalence. Some of these have already been identified, some of these genes, in relation to waist-to-hip ratio, which is a major medically important and relevant trait, so the ratio of waist circumference to hip circumference. But some of these are novel and haven't been identified before. So are we just picking up waist-to-hip ratio? And it doesn't seem like we are. It seems like we're picking up something different. We're not just picking up waist-to-hip ratio, because if you look at the lipedema cases in green there, you can see that they, on that distribution of waist-to-hip ratio, they're not on the extreme of waist-to-hip ratio. They're somewhere in the middle. So hopefully we're not just picking up waist-to-hip ratio, because we want a more refined phenotype than just waist-to-hip ratio. We can also look at not only the SNP as the unit of analysis, but also the gene as the unit of analysis. So looking at the gene overall. So a gene is just like a chunk on a chromosome or in our DNA, and looking at all the variants in that gene in relation to lipedema. And so we've identified some of the same genes that were previously identified, but also some other ones that weren't identified, such as the HOX-A genes. So another interesting follow-up analysis that we can do is ask the question, when we look at all the genes that we identified, where do these genes seem to be expressed? And there's publicly available data looking at where genes are expressed. What parts of the body are they expressed? And it seems like these genes that we've identified are expressed mainly in the subcutaneous adipose tissue, as you would probably guess and hope for, and then as well as breast mammary tissue. So we're going to follow up on these and look at the genes individually to see where they might be expressed. And this is kind of a long road, because a lot of the genes that we've been identifying in these studies, we don't really know what they do. And so it's kind of a long road to figure out what these genes are doing and where they're doing it and how they're doing it. So in terms of next steps, we're going to try other ways of defining lipedema and different prevalence assumptions. So trying a prevalence of 1%, trying a prevalence of 10%, maybe trying a prevalence of 40%. There is one study that found that the prevalence of lipedema in Germany, I think it was 40%. So we'll try that. And then importantly, can we replicate these genes in other studies with diagnosed lipedema patients? So our phenotype is quite crude, but if we can replicate this in other studies, that'll be really give us more confidence in the results. And then we're also going to be collaborating with Karen Herbst and looking at these genes that we've identified and ask, are these genes differentially expressed in the thigh tissue of lipedema patients compared to the thigh tissue of controls? And then we also want to know, ultimately, in order to be able to understand the disease and figure out ways to treat the disease, we want to figure out what biological pathways are pointed to by these genes. And so just a few bottom lines. I don't think there's one gene for lipedema. I might be wrong, but I don't think there is one gene. I think there are many genes, probably at least dozens, probably maybe hundreds. And I think we're going to find the genes in the next 5 years. But I think what's going to take longer is figuring out what these genes are doing. All right, well, I'd like to thank the Lipedema Foundation for funding to do this research, as well as collaborators, including Dr. Herbst. Thank you, and you can contact me there.